Hola, muy buenos días. Sean bienvenidos todos ustedes a la 40ª edición del Coloquio de Investigación de la FES Iztacala. Estamos en nuestro segundo día de coloquio y tenemos la segunda conferencia magistral que va a ser impartida por el doctor Llye Chen. Le cedo la palabra al doctor José Pablo Romero López, quien hará una pequeña introducción sobre el doctor. Adelante, doctor.
Muchas gracias, doctora. Voy a empezar en inglés y voy a presentar al doctor. So, good morning, everyone, and thank you for being here. I will introduce you to Dr. Llye Chen, who is our speaker today, and who very kindly accepted to share some of his research with our university. So, very briefly, Dr. Llye Chen developed his interest in immunology research during his PhD in the University of Oxford from 2009 to 2013, during which he discovered functional explanations for some genetic findings in a type of arthritis called ankylosing spondylitis. This included ERAP1 and HLA-B27 epistasis, HLA-B position 97 variations, and he also helped to define the transcriptomic features that are unique to T cells from ankylosing spondylitis patients. From 2015 to 2019, Dr. Chen worked as a senior postdoctoral fellow in the Ultra-DD Consortium to identify and validate underexplored novel targets in AS. Dr. Chen started his own group in 2019 at the University of Oxford in the Bodnar Research Center with a research focus on the discovery of novel immune regulators and drug targets in human immune-related diseases. To this end, his group has developed a unique strategy to integrate genetic and patient omics datasets to discover new biology followed by CRISPR or siRNA-based functional validation using human primary immune cells. So thank you very much, Llye, for accepting being here. And this room is yours now. If you can please share the screen and start the presentation.
Okay, thanks a lot for the kind introduction, Pablo. And yeah, firstly, very good morning, everyone, and I'm so glad that I got the opportunity to share some of our work with you. So as you can see from the topic, the title actually, what we do is mostly trying to discover novel disease biology in the context of the inflammatory arthritis, with a special interest in spondyloarthropathy, I think let me make this slightly smaller. Yeah, that will work better. So to start with, I would like to talk a bit more about my work as a postdoc. So at that time, I was really exclusively working on this ankylosing spondylitis, a form of the chronic and inflammatory arthritis, mainly affects spine and pelvis, but apart from that, patients do have some extra-articular symptoms, including the inflammation in the gut. Let me get my laser pointer. Inflammation in the gut or skin. So this is actually a patient with both of the spinal arthritis and the psoriasis. And some patients will develop uveitis, especially in the older population. And the other, a smaller group of the patient even have lung fibrosis. And so at that time, the biology or pathology of ankylosing spondylitis was not very clear, and then probably still not very clear now. But when I joined the field, there was actually a sort of big progress in the genetic work in the field of ankylosing spondylitis. Several big GWAS studies were carried out, identified at that time, I think was almost 100 genetic locus affecting the risk of developing the disease. And then one of the gene that was actually sort of the focus of the whole field at the time was a gene called ERAP1. And this is sort of an aminopeptidase that's located in the endoplastic reticulum, and it's very important for the presentation of MHC class 1 peptide. And then because of the its strong genetic association with the ankylosing spondylitis. And also very importantly, and also quite striking at that time, was that the association of the ERAP1 with ankylosing spondylitis is only present in the individuals that are positive for HLA-B27. So this kind of very sort of a unique epistatic association highlight that it's very likely that ERAP1 and HLA-B27 actually work together in the contribution to the pathology of ankylosing spondylitis. And so because of that, I was working on the potential regulation of HLA-B27 function by ERAP1. And then later, because apart from HLA-B27, other HLA-B alleles were also actually associated with the increased risk of developing ankylosing spondylitis. And the HLA-B51 is one of these alleles that was also associated with the risk. And because of that, we also have a study the function role of the ERAP1 in the presentation and expression of HLA-B51. And then last sort of project in the context of HLA-B and ERAP1 was that in a genetic study published, I think in 2016 or 15 in Nature Communication, shows a very unexpected and striking finding, which is instead of HLA-B27 itself being associated with AS, actually it's the variation of the amino acid at position 97, and that's even more important for this kind of genetic risk. And because of that, at that time we studied the impact of the genetic variations at this position and its on the expression of the HLA-B alleles. And then apart from the sort of the function of AS associated genes, I was also involved in sort of the unique immune cell phenotype in this form of the inflammatory arthritis. And we found that in the TS17 cells, which is a key pathogenic T cell subset in AS, and then the cytokine produced by TS17 cells, and then the interleukin 17A is actually a very clinically proven drug in treating the patient with ankylosing spondylitis. So back in 2016 or 17, we found that the TS17 cells from the patient with AS, a novel microRNA called microRNA 10B is actually hugely increased in the TS17 cells from the AS patient, and then we found that actually this increase is because of the enhancement of the inflammation in myeloid cells that can produce higher level of the pro-inflammatory cytokines in supporting the upregulation of this microRNA in TS17 cells from the patient blood. And then later we also found that actually this microRNA, instead of promoting TS17, is actually anti-TS17 microRNA regulator. So it sort of almost worked as a sort of checkpoint microRNA that were upregulated to suppress the TS17 response. So that was really interesting. And then around a similar time, I was also helping one of the colleagues, Llye Chen, to actually study a subset of the T cell in the that are expanded in the SPA, and this subset of the T cell express higher level of the GM-CSF and also express high level of a protein called GPR65, and then we found that this GM-CSF positive lymphocyte are increased in both of the blood and synovial fluid of the patient with the PSA, and we also found that interestingly, the SRA knockdown of this GPR65 gene was able to downregulate the production of the GM-CSF by T cell. what's worth mentioning is that GM-CSF is a very, very important cytokine for the generation of the pro-inflammatory M1 macrophage. And then lastly, I was very, very interested in sort of translating our findings to a sort of patient benefit. And then and because of that, I was involved in 2 translational projects. One of them exploring the possibility of the JAK inhibitor in suppressing TS17 response, and then and we essentially found that JAK inhibitor was a very, very potent a tool to suppress TS17, and we also showed that the siRNA knockdown of the JAK kinase was able to suppress the T cell proliferation and then the TS17 response as well, and because of that actually, and targeting JAK could be a very, very nice target and a nice approach to treat the patient with the ankylosing spondylitis. And then we were quite pleased to actually see that over the last few years, JAK inhibitor got approved in treating several forms of inflammatory arthritis, including rheumatoid arthritis and ankylosing spondylitis. And it's so nice to actually see some of the more basic and really preclinical kind of work support and helped in the development of the treatment that's beneficial for patients. And then in at the same time, we also were interested in the epigenetic regulation of the T cells in a inflammatory condition. And for this project, we screen a library of epigenetic inhibitor using the CD4 from patient with ankylosing spondylitis, and then we found a family of the bromodomain containing a regulator called BRPF. There are actually 3 members in this family, and then we found that inhibitor targeting this family were able to suppress the T cell inflammation. And then later we performed the siRNA screening on all the 3 members in this family, and then we found that one of the member was not affecting the T cell inflammation, and then the second member in this family, following the either CRISPR knockout or the siRNA knockdown, suppress the pathogenic function of the CD4 cells, reducing through the reduction of the IL-17 production, GM-CSF production, and then the last member of this family, interestingly, following the siRNA knockdown or the CRISPR knockout, enhance the proliferation of the CD4 cells and also enhance the pro-inflammatory feature of the CD4 cells. And so this is a project that's still ongoing in the lab. It's probably one of the oldest project in the lab, has been going for probably 7 or 8 years. And so hopefully we can finish this project sometime. And then I would like to just firstly just give you an outline about my previous work, but now I'm going to show you some of the key figures related to different aspect of the work, and about the regulation of ERAP1 towards HLA-B function. At that time, what was really interesting was that this kind of epistasis between ERAP1 and HLA class 1 allele was not only discovered between HLA-B27 and ERAP1. It was also discovered in HLA-B51 and ERAP1. And then apart from that, the similar sort of epistasis was also reported in psoriasis for the HLA-C06 and ERAP1. And so you can see that actually this is the 3 publications coming out sort of around a similar time. And then because of that, everyone was sort of thinking that, well, it must be some of the shared or common mechanism that ERAP1 utilized to regulate this different subtype of HLA, and this type of a regulation must somehow utilize similar pathogenic pathways in contribution to the autoinflammatory disease, because all 3 conditions can be grouped into autoinflammatory disease. And then And where we started from was actually firstly generate a crystal structure of the ERAP1. And then we were focusing on one of the very important coding change, which is actually providing a protective effect towards the development of the AS. And then we found that the ERAP1, wild type ERAP1 have two states, either open states or closed states. And then when it's open, then the substrate of ERAP1, which are normally long peptides ranging between 10 to 16 or 18 amino acid long, and they can actually get loaded in this area. And then the ERAP1 can start to chop down those peptides to a shorter length. And then normally the chopping efficiency went down dramatically when the length of the peptide go down to about 9 to 8 amino acid long. And that's because this is the length that's actually optimal for the complex of the formation of the MHC class 1 complex. And then, and importantly, this mutation is actually located in this yellow domain, which is served as a sort of hinge, affecting the closed and open state of the ERAP1. And then, what we did at the time was to carry out some of the in vitro peptide digestion experiment, comparing the function of the wild type ERAP1 versus this protective ERAP1 variant. And then what we are showing here is essentially that this protective ERAP1 variant trim the peptide substrate much slower than the wild type ERAP1. And then because of that, it's sort of supporting that the loss of function ERAP1 or the genetic variant led to the reduced expression level of ERAP1 might actually be protective in AS. And if that's the case, then ERAP1 inhibitor might actually serve as a very, very good potential therapeutic approach to treat the disease. And built from there, then we move on to answer the question that, well, if ERAP1 is important for the HLA-B27 function or not. And then to do that, we utilize the siRNA to silence the endogenous ERAP1 using a cell line overexpressed HLA-B27. And then we firstly want to know that whether the silencing of ERAP1 using this shRNA on this second bar here, ERAP1 shRNA, whether this kind of silencing affect the composition of the peptide that are presented by HLA-B27. So what we found was that following the knockdown of ERAP1, the percentage of the NIMER essentially means the peptide that are actually composed of the 9 amino acid, and this is the most favored length of the peptide by MHC class 1. So percentage of the NIMER were actually reduced in the ERAP1 knockdown condition, and then in contrast, those longer peptides, especially the peptides that are made of 11 or even 12 amino acid, so 11 or 12 they become more abundant in the absence of the ERAP1. So this data sort of suggests that ERAP1 seems to be very, very important for the generation of the optimal peptide pool for the formation of MHC class 1. And then in here it's actually HLA-B27. And if you knock down the endogenous ERAP1, and then it seems to be actually facilitating the presentation of those longer peptides. And then we also carry out an experiment and trying to answer the question that whether the knockdown of ERAP1 can indeed affect the ability of the antigen presenting cells to activate the T cells. So here we use a sort of a Alice spot kind of experiment in which we co-culture the HLA-B27 expressing cells either with endogenous ERAP1 or with reduced endogenous ERAP1. We co-culture those antigen presenting cell lines with the human CD8 T cell clone that is specific to one of the HIV epitope, and then in the graph in the sort of graph below, each spot that you see is essentially a T cell that have been producing interferon gamma in response to the presentation of the peptide by the antigen presenting cells. So what was clear was that following the silencing of ERAP1 in the antigen presenting cells, there was a reduction of the activated T cell clones that produce interferon gamma. So this data essentially suggests that ERAP1 is actually quite important for the presentation of the viral epitope, and then the knockdown of the ERAP1 was able to compromise the ability of the antigen presenting cells to activate the T cell. And then but actually all these findings do not directly, it doesn't really completely tie into the pathology of the ankylosing spondylitis. And this is a very nice review came out from Paul. And then so and this graph, this cartoon very nicely summarized the three main hypotheses in terms of the H2A B27 pathology in ankylosing spondylitis. So if we look at the A, it essentially shows that the H2A B27 expressing antigen presenting cells under stimulation. This stimulation could be, the product from bacteria, and they can actually induce this kind of stimulation can induce the expression of the H2A B27 homodimer, and then this kind of homodimer are actually expressed on the cell surface, and then this cell surface homodimer can interact with the T cell through the immunoregulatory receptors, such as KIR3DL2, and then this kind of interaction can then enhance the expansion of the TS17 phenotype, and then so because of this sort of hypothesis, at that time we wanted to know that whether ERAP1 might be affecting this kind of the pathology. And so to answer this question, we firstly want to know that if we knock down ERAP1 in the B27 positive APC, does it alter the expression level of the B27 homodimers on the cell surface? And then secondly, if that's the case, we want to know that whether the regulation of the B27 homodimer on the cell surface and can it indirectly affect the T cell inflammation? So to answer those questions, we firstly use both of the ERAP1 siRNA or the ERAP1 inhibitor to treat the cell lines expressing the H2A B27, and then we utilize two different antibody. The HC10 is antibody that recognize the beta-2-M3 MHC class 1 heavy chain. And what's worth highlighting is that HC10 is not actually selective towards H2A B27 free heavy chain. It also binds to the MHC class 1 and class 2s as well. And then, but there are evidence suggesting that the HC10 reactive molecules are increased in the cells from patients with AS, and then that's especially the case for the monocyte. And ME1 is an antibody that can recognize the classical complex of the H2A B27. But again, it's not perfect because they also react to other H2A-B alleles such as H2A-B7. And so because of that, actually, we need to treat these findings with a pinch of salt. And then, but this is only reagent that were available at the time. And actually, I think this is probably the only reagent available now as well, because it's really difficult to develop an antibody that's actually selective for H2A B27, given that often the H2A B27 only differ from other HLA-B alleles for a few amino acids. And then, because of that, we after we observe the reduction of the HT10 reactive HLA-B27 free heavy chain by the ERAP1 silencing or by the increasing concentration of the ERAP1 inhibitor, and then we were pleased to see that the classical HLA class 1 complex, especially HLA-B complex, were not affected by either ERAP1 silencing or the ERAP1 inhibition. So that sort of give us a bit more confidence about the finding. And then after that, importantly, we definitely want to know that whether the alteration of the HLA-B27 expression on the cell surface affect the TS17 frequencies or not. And this is actually an important experiment to bypass the limitation of the antibody that we have just discussed. And then so here I think we use the two different techniques. On the left-hand side is the intracellular cytokine staining, looking at the frequency of IL-17A producing cells in the total CD4 population. And then on the right here, it's the production of the IL-17A cytokine in the supernatant, so measured using ELISA. So so you can see that for either using intracellular cytokine staining technique or using the ELISA, we observe a similar and consistent result, and namely the downregulation of the TS17 frequencies following the ERAP1 knockdown in the APC, and similarly when we use inhibitor, we observe similar result. So that was quite exciting and quite nice. And then actually following this publication, we knew that quite a few farmers were actually putting the resource into the generation of the ERAP1 inhibitor with the aim to developing new drugs to treat increasing spondylitis. And both of the GSK and Johnson & Johnson were interested. But I think they stopped the project. I think both companies stopped those drug target discovery drug development program because the concern of the ERAP1 inhibitor in affecting the thymic selection of the T cell. And then, but this is a topic that I'm not going to go to too deep today. But in summary, this kind of data seems to suggest the ERAP1 could be a very good drug target, but because of the safety reasons, the translational development of the ERAP1 inhibitor was not carried out in the end. And then, so in summary, we found that the ERAP1 seems to be very important for the HLA-B27 function. We found that firstly, it affects the HLA-B27 peptidome. So peptidome essentially means that all the peptides that can be actually presented and bound by HLA-B27. And apart from the peptidome, we also found that the ERAP1 is actually important for the presentation of the HLA-B27 restricted peptide to the human CD8 T cell clone. And then lastly, we found that the ERAP1 can regulate the expression of HLA-B27 free heavy chain expression on the cell surface, and then this kind of the regulation indirectly act on the TS17 cells. And then move to the sort of Behçet's disease for the to discuss the sort of ERAP1 and B51 project. So Behçet's disease is sort of a bit similar to ankylosing spondylitis in the sense that it's also a systematic inflammatory disease. But actually it has the organ that was most affected by Behçet's disease is actually eyes. So it's traditionally seen as an ocular disease. but the patient do also have the inflammation in other organs, which include the arthritis, skin, and then this is quite interesting because as I mentioned earlier, ankylosing spondylitis patient also often have the inflammation in eye and in skin as well. So even thinking about the clinical symptoms, there seems to be some overlap between these two clinical conditions, and then this actually this make the make us believe even more that it's possible that a shared pathogenic mechanism might be present between AS and BD, and the ERAP1 and HLA-B could well be at least one of those mechanisms. And so I'm going to only summarize the data using one slide. We did look at the impact of the ERAP1 on HLA-B51 expression, but we found the result was not very consistent. It seems to be cell type dependent. And like I remember that in B cell line, the knockdown of the ERAP1 upregulate the HLA-B51 free heavy chain expression. But in epithelial cell line, we saw an opposite result. So because of that, we didn't really think that we didn't feel like we're confident enough to say that we have already found the answer to this question. But instead, what we found to be very consistent and very clear was the impact of the ERAP1 on the HLA-B51 peptidome. And then, so at the time, we were not very happy about the fact that we don't really have a good antibody that can selectively isolate the HLA-B51 complex. And then to overcome that problem, we generate a sort of a triple knockout cells in which we sequentially knock out HLA-A and B and C, and so that this cell line doesn't really have any of endogenous MHC class 1 or classical class 1 molecules, and then we put HLA-B51 back into the cells, and then using this approach, we built up a system that is a sort of very clean system that only the MHC class 1 and HLA-B51 are present, and then using this kind of the cellular model system, we were able to isolate very pure MHC HLA-B51 complex, and then we compare the peptidome of the wild type cells versus the ERAP1 knockdown cells, and what we found was slightly surprising to us. That is that we firstly found that in addition to two sort of classical HLA-B51 subpeptidome, which is proline, either have proline and arginine in position 2 or have alanine at position 2. But the peptides that don't have either proline or alanine at position 2 hasn't really been actually appreciated for the HLA-B51 peptidome. But we found that actually this population is definitely present because we use a very clean system. We know that all the MHC class 1 complex that we eluted can only be from HLA-B51. And so we're confident to say that actually this population of the peptide do exist, and they indeed can be actually bound by HLA-B51. And then importantly, following the knockdown of ERAP1, we observe a very clear downregulation of the proline 2 peptides and the increase of the alanine 2 or the non-proline or alanine 2 peptides. So this was very, very interesting. And then, but apart from that, we also actually look at the length of the peptides that are bound to HLA-B51. And because we thought that it's possible that these three subpeptidomes actually might have different features, and then we feel it would be better if we perform the length analysis for the individual subpeptidomes. And then so this is what we did. We found that actually for proline 2 peptides, we didn't really see much change, but in contrast, in either the alanine 2 or the third population, so the unconventional population, we observe a very clear downregulation of the shorter peptides and then upregulation of longer peptides. And if you remember, actually, this is something that we observed for the ERAP1 and HAB27 project. So this is rather interesting. I think it almost suggests that the regulation of the HOAB pathology is very likely actually mediated through the changing or altering the length of the peptide that are presented by the HOAB. So in this case, it's B51. And then things like the knockdown of the ERAP1 consistently reduce the length of the shorter peptides and increase the percentage of the longer peptides. And sadly, we still don't know exact biological mechanisms behind those observations. And I think currently a few sort of hypotheses have been proposed. One of them being that maybe the either HOAB27 or HOAB51 that has been actually loaded by longer peptides, they might be a bit less stable, and then as a result, they might actually lose the peptide a bit quicker, and then they will actually contribute to generation of the beta-2-M3 and the beta-peptide 3 heavy chains being expressed on the cell surface. But this is a question that haven't been completely understood and explored. And then another important limitation of all this work that I've carried out for both of the HOAB27 and B51 was we were limited by the material to work with. So all this work will carry out using the cell line, and we don't really know whether in primary cells, especially in primary cells from patient, whether all those findings will stand true. And then this is some of the future work that will definitely need to be carried out. And then actually in our lab, one of the students continue to pursue all those questions using the primary cells from the HOAB27 positive AS patient. So hopefully in 1 or 2 years' time, I'll have the chance to share with you about those stories. I might skip the HOAB51. I might skip the HOAB position 97 part for the benefit of time, because I realized that I probably have about 45 minutes left. And I would like to maybe switch the gear slightly and then start to talk a bit more about the immune function, and then talk about the T cell, one of my favorite cell types to work with. And then so this is a bit of the background. I think the reason that we got interested in the TS17 cells and also the GM-CSF positive CD4 was because when you look at the frequency of those cells, TS17 on the left and the GM-CSF positive cells on the right, you can see that they are really selectively, specifically upregulated in the patients, SPA patients, and even when you compare this group of the patient with rheumatoid arthritis, there's a very, very clear increase as well, and suggesting that this kind of the increase in its cell number is not due to the general inflammation, and it seems to be actually specific to the SPA. And then something in line supporting this is that the blocking of IL-17A in rheumatoid arthritis has failed in a clinical trial, so highlighting that TS17 cells are probably not important, at least for most of the patients with rheumatoid arthritis. And then so we were looking at the different subset of the T cells, and then at that time, because GM-CSF positive CD4 cells were actually less well studied, and so we want to actually identify the key sort of the regulators for this subset of the cells. So here actually this is mostly Hussein's work. He sought out this 5 different population of the T cells, including naive T cell, interferon gamma producing T cell, GM-CSF producing T cells, IL-17A producing T cells, or the GM-CSF and IL-17A double positive cells. And then when he carried out this work, then he found that GPR65 is actually very selectively upregulated in the GM-CSF positive population and the GM-CSF and IL-17A double positive cells. And as I said earlier, that when we carry out the siRNA knockdown of the GPR65, we observe a downregulation of the GM-CSF. And then interestingly, we didn't observe a downregulation of the IL-17A. And this data is actually in contradictory to what was published. I think that was 2 years before that, before this work, in mirroring models, in which they show that GPR65 is very important for the generation of the pathogenic TS17 cells. And then, but actually, this is actually slightly outdated now. Llye Chen and also Paul, who was Llye Chen and my DPhil supervisor, PhD supervisor, sorry, DPhil is Oxford and Cambridge thing. Paul and Llye Chen together, they actually were able to got enough fund and support from pharma to carry out a phase 2 clinical trial. And then this actually trial have been completed, but the result was not really positive. You might see some of the data being presented very soon. I think one of the, you probably have even presented the outcome of the trial half a year ago in ACR. But actually, currently a student in Paul, working with Paul and Llye Chen, she's actually trying to understand that if she compare the responder and non-responder to the GM-CSF blockade, whether there are some specific feature affecting the response to the GM-CSF blockade. So sorry, I should firstly say that actually it's not complete failure because some of the patients do respond to GM-CSF blockade, but not all of the patients responds to GM-CSF blockade. So this could hopefully due to the heterogeneity of the patient population. So you can see that actually lots of our work is actually quite translational focus, and often some of our I think we are trying very, very hard to translate the findings from the basic science to the sort of clinical application, and then most importantly to the patient benefit. And so this is about the microRNA project. As I said earlier, that we compare the microRNA expression profile between the TS17 from AS patient versus TS17 from healthy control. And then we only got limited hits, and then one of the hits that we highlighted here is the microRNA 10b5p, it's highlighted here. And then we confirm this finding firstly using the qPCR, and definitely this microRNA is upregulated in the TS17 cells from AS patient. And then to understand the biological role of this microRNA in a TS17 response, we overexpress this microRNA using its mimic and in the primary human CD4, and then we found that the overexpression of microRNA 10b downregulate the IL-17 production by primary CD4 cells from patient. And then for anyone who work with microRNA, you will always be puzzled by a question that's what is the target of the microRNA? And this is often a very challenging task because lots of microRNA has more than one target. And then, but we had a practice in doing that, and then by comparing the transcriptome of the CD4 cells that's been transfected with a control microRNA versus the microRNA 10b, and then we're actually quite lucky that out of the bulk RNA sequencing, we found at least one of its target, which is MAP kinase. And again, this is a MAP kinase. This is one of the target that has been attracting some of the interest from pharma to develop it as a drug target. And so what we firstly carry out was to confirm using experiment that whether the overexpression of microRNA-10B can indeed suppress the this kinase MAP3K7. And then the qPCR results suggest that's indeed the case. It's very, very clear that the expression level of the MAP3K7 was substantially downregulated following the overexpression of MIR10B. And then we also confirm its impact on the IL-17 production, and then essentially the siRNA knockdown of this target can actually mimic a phenotype and that MIR10B delivered. So essentially to sum up for this project, we essentially actually start from unbiased sequencing practice, and we found the microRNA-10B is actually specifically upregulated in TS17 cells from AS patient. And then later, we again through the RNA sequencing experiment, we identified a target of the MIR10B, and then we confirm that this target of the MIR10B is actually a very important kinase in a TS17 response. So now actually, I think I spent almost 1 hour talking about my postdoc work, and I think I should definitely be keen to share some of the work that we have been carrying on since I started my lab about 3 years ago. And so I think one thing that I learned from all my postdoc work was that it's very, very clear that genetics and omics-led functional research is really, really powerful approach in identifying new biology and new drug target. And because of this, when I started my lab, I wanted to actually achieve the following goals. And then two of the main goals is to firstly to really advance our knowledge in human immunology, and especially in a disease setting. And this is I think this is really important because it's often really, really hard for us to provide a treatment or novel treatment for the patient that do not respond to any of the current drug. And then I think the main reason that is so difficult is actually not because we haven't got enough candidate drug target, but it's often because the biology of target is not clear enough. And I think one of the really, really good examples is actually the blocking of the IL-23 in ankylosing spondylitis. Because IL-23 has a very, very strong genetic support, because IL-23 receptor is a genetic risk for the development of ankylosing spondylitis. And then IL-23 are well known to be a very important upstream cytokine of the TS17 cells. And blocking of the inhibition of the TS17 cells has been, effective in treating patients with AS. And then you expect the IL-23 blocking should work in treating the patient with AS, but somehow clinical trial failed. And then I think till now, I don't think anyone knows why. And I think this has highlighted that its importance of understanding the biology in the development of the new drug options or new drugs to treat the human disease. And another thing that we feel is really has been sort of largely missed especially in the human immunology study is the crosstalk of the different cells. And for people who are sort of lucky enough to work with mice and with mirroring models, I think they sort of have less of this problem because those crosstalk between different immune cells are naturally present in an in vivo model. But in vitro, lots of lab, including actually, including myself when I was working as a postdoc. All this project, I work on one cell line at a time, or work on one cell type at a time. And then we rarely actually use a sort of a cellular model that actually include more than one cell type. And then I think there's a reason behind this, because it's really difficult to actually generate those cell crosstalk model, and largely because we don't really know enough about the cellular composition of the different cells in human body, and then certainly in human disease as well. But I think this kind of all these obstacles have been actually partially overcome by the development of the single cell RNA sequencing, and especially the spatial single cell RNA sequencing. And because this kind of the techniques actually allow us to really have a single cell resolution about the cellular composition of a human of an inflamed joint, or of an inflamed eye, and then this data has actually become largely available for most of the human disease now. Actually, I think if you look for the inflammatory, at least in the inflammatory disease, almost for any disease, you can actually find some data set using the single cell. Of course, some of the disease might actually have a higher quality of the data set, and also have a higher abundance of the data set. And then especially like in the context of the arthritis, the rheumatoid arthritis have a huge single cell RNA sequencing data set covering T cell, myeloid cells, fibroblast. And then in other conditions, maybe the tissue data set is not quite available yet, such as the psoriatic arthritis. This data set is actually hasn't been published yet. But at least I know that actually quite a few labs in the field have already been actually generating this kind of data, and then very likely that in 1 or 2 years' time we might actually see this kind of data become available. And then any lab in the world with interest in the disease can actually have access to those data set, and then we can actually build on those data set, and then to actually increase to build up the in vitro model system that can hopefully actually mimic, at least partially mimic, what's going on in an inflamed joint, for example. So apart from the first part, the second part, you can tell from my talk that I really have a heart of the translational medicine, and then we really want to actually to generate the knowledge that can be actually useful for clinics, and then because of that, we are more sort of human centric, and we think that mirroring models are very useful to model the in vivo scenario, and then to, as I said earlier, to model the to have the cell crosstalk in the game and in the model, but actually to understand to solve a human disease, and we have to rely actually more on the on human cells, and so we have been spending lots of time trying to build up different models to mimic the information that's going on either in inflamed disease or in the cancer as well. Cancer is another direction that we are working on, but we do so solely using the human primary cells, because we know that actually this very big difference between the primary cells and cell line, and then cell lines definitely have its own advantage, but frankly, a lot of the findings using the cell line, including the work that I just presented to you earlier, my own work, do I completely trust it? I found it's hard for me to say that I completely trust my own data, because they'll carry out in cell line. I really don't know that whether that will be all those findings will stay true in the primary human cells, and then so why if you cannot completely trust it, then why not just directly, why not just start from the primary cells directly, and then just don't use those cell line work at all, or at least start from the primary cells and then only use the cell line for some of the sub questions that's actually less relevant to your primary questions. And then, but as I said, mirror model is still useful. But I think we can actually rely more on the primary human cells and then using the 2D or 3D of the disease model, including the organoid models, and we use those to actually answer the main questions to build up to test or optimize the main biological story. And then for the in vivo kind of the work, we can then actually use mirroring disease model to test those secondary questions. So at least this is the approach that we believe to be a good approach for a translational purpose, and then for the advancing of the human immunology. And this is the approach that we decided to take. And then in terms of our strategy, and I think we rely probably we can sort of separate our work into two parts. And then the first part is really to identify the sort of the new biology or new potential drug target, and then for this part we utilize three elements of the sort of tools. And then the first tool is using the disease or immune response specific gene signatures, and this kind of signature comes from any kind of omics, could be actually proteomics, could be actually the bulk RNA sequencing, could also be single cell RNA sequencing, could also be single cell DNA sequencing. And then apart from this, we definitely think that genetics risk from the GWAS provide a very, very useful tool for us to identify the not only new biology, but the new biology that's relevant to the disease. And then lastly, we thought it's really, really important to have a approach allowing us to actually validate to firstly screen for and then validate the novel immune regulators that we found in primary human cells. So this is the sort of the first part. And then after we identify those either novel immune regulator or novel biology, and then it goes without saying that we need to validate it. And then following validation, then actually we can actually carry out both of the mechanistic study and the translational development. And I would like to for this, I would like to give you some of the example projects. We have lots of projects going on in the lab, and then I think the project I'm going to show you probably is the one that has gone further than the other project. And then using this this project, I think we start from genetics and then omics data, and then we identify a novel immune regulator, and then we actually follow it up by trying to identify a new FDA approved drug that can actually be repurposed to treat this target. So sort of go all the way from the first part to the target validation, and then to the translational development. So hopefully, actually, the example that I'm going to show you later can actually convince you that our approach is actually reasonable and it's working. But before that, I would like to just highlight it that a few of the data suggesting the power actually from GSK, showing that the drug target with genetic support can actually double the chance of the success in clinical trial. I'm not going to go to details because this involves lots of the very dry kind of data analysis. And then this finding was then actually validated by a second study, again from a company, AbbVie, and essentially showing a similar result. And then I might skip this because this is maybe a bit too genetics for the audience. And then but essentially for one of these sort of the key research program, we're trying to identify the immune regulator in the context of inclusive spondylitis. And then the approach that we utilized was to actually build on the 113 genetic locus loci that have been found to be associated with AS. And then we use some functional genomic data set, mostly use the expression quantitative trait locus, which essentially tell us the association, whether a genetic variant affect the expression of a certain gene. So this is one of the example that for the individuals that has GG, those individuals actually has a much lower expression level of this gene STEP1 in comparison to those carrying AA. So there are 120 individuals in the AA group, and then there are 44 individuals in the GG group. And then also importantly, for the individuals that are sort of heterozygous and have a copy of A and a copy of G, they actually expression level of STEP1 sit between these two populations. And so this is sort of a typical EQTL kind of analysis. And then we essentially utilize this kind of the data set to help us to identify the genes that are actually affected by this genetic risk discovered in GWAS. And then, so for this project, we using this approach, we have identified quite a few of T-cell regulator. And then as I said, that I will just showcase one of the project that is about a gene called ERN1 encodes the protein called IRE1. So we firstly, so this project was mostly contributed by a postdoc in a lab, Dr. And then, so she has been, should I admit? I can't do it. I'll leave it to the tech support. So in this work, we were actually we screen a multiple sort of GWAS genes identified using this approach, and then we started from a list of the GWAS candidates, and then we asked the question of whether any of those genes are differentially expressed between some of the important subset of the T cells that are important for inflammation. And here we focus on Treg, TS17, or the GM-CSF positive CD4s. And then we found that this gene ERN1 was not only upregulated in the TS17 cells, but also actually upregulated in the GM-CSF positive cells. So because of that, at that time, I think we started this project maybe four years ago at least. So at that time, we thought that both of the GM-CSF positive CD4 and TS17 are very important. So we thought, oh, we have to actually follow this gene. And then, and that's what we did. So this gene, the genetic variant link us to this gene is a single nucleotide polymorphism. We normally call it SNP. So this SNP is actually located within intron of the ERN1 gene. So this is ERN1, and then this is this is the promoter, and then you can see that the small bar there, they are actually exon, and then the gap between the black blocks, they are intron. So this SNP is actually located in this intron. And then using the EQTL data set I mentioned earlier, we found that actually in addition to TS17, this genetic variant also is also associated with the expression of ERN1 gene in monocytes as well. And then because of that, we thought that, well, in that case, maybe we shouldn't limit ourselves to TS17 cells. We should probably also consider the possibility that this gene is also affecting the immune function of the monocyte. And that's what we did. And then I will skip this. So now we actually look into the neurobiology of the ERN1, and then we found that the protein encoded by ERN1 called IRE1 is actually quite an important a sensor of the ER stress. And then following the sensing of the ER stress, IRE1 can lead to the splicing of the transcription factor called XPP1. And then following the splicing, the XPP1 gets activated, and then it becomes a sort of an active transcription factor. And then it can get translocated in the nuclear and then perform the transcriptional regulation. And then so what we were proposing at the time was that because the only two clinically approved treatment at the time was the blocking antibody against TNF-alpha or the blocking antibody against IL-17A. As I said, the IL-23 failed, although it's upstream of the IL-17A. And so we wondered that, well, is it possible that that if IIE1 is important for both of the T cell inflammation and myeloid cell inflammation, and then is it possible that if we block this gene, can we potentially achieve the blocking of these two pathways at the same time? It's sort of a one stone and two birds kind of a strategy. And then so that's why we went to test. So we firstly used siRNA to knock down this IIE1 protein, so ER1 gene, in the primary human CD4. And then so on the graph A shows the efficiency of the knockdown. In primary T cells, it's really hard to achieve very high level of the knockdown efficiency using siRNA. And then 50% is actually quite a good knockdown efficiency already. And then and despite only 50% of knockdown efficiency, we actually observe very a clear downregulation of the IL-17A. And then apart from that, some of the cytokines, especially IL-17F, has also been downregulated to a similar level. And this is suggesting that the IIE1 siRNA seems to have been targeting some upstream pathway that are actually controlling both of the IL-17A and IL-17F production. And in contrast, the IIE1 siRNA has weaker effect on interferon gamma and GM-CSF. But I think this could be a numbers game. If we increase a number, it might be significant as well. But it's very clear that the impact of the IIE1 siRNA on the 17A and F were way more striking, way bigger than its impact on the interferon gamma and GM-CSF. So, and we also carry out similar work in the monocyte and macrophage. So on the graph A is monocyte, on the graph B is macrophage, and then we also perform the siRNA knockdown of this target, and then we observe the downregulation of both of TNF-alpha and IL-23. Sorry that I only show n equals 2 here, but actually this figure is quite dated. I think we should have at least n equals 7 or 8 now. But the result was very clear. So the result was really good, but with our experience with ERA1 inhibitor, as I told you earlier, we were really thinking about whether targeting IIE1 or an inhibitor for IIE1 is it going to be a really good drug option or not? And then because IIE1 actually has been shown to be important in multiple biological process, including like the maturation of the B cell and including the function of the NK cell as well. And because of those, we're really worried that an inhibitor for IIE1 might actually be sort of behave a bit more like immune suppressive kind of the drug. And then that's probably not something that we want for patient, because the side effect will be too severe. And then we try to actually bypass this limitation. And one of the approaches that we try was to screen some of the FDA approved drugs that are known to be potentially affecting the pathway. And we screen quite actually we didn't screen too much, and then probably we're lucky here, and then we were actually able to identify a drug, and then this drug is sort of a generic drug that was developed to treat a rare genetic disorder. It's a very old drug, and it can be taken orally and with mild adverse events. And then and sorry that the reason I put I could not share the name of the drug is because I think we are still discussing with the tech transfer office in Oxford University, and we have. It's unlikely that we will generate any IP around this, but but I was advised not to reveal the name yet, so apologize for that. And then and then I would like to show you some of the data that we have generated using this drug. And then so firstly we want to, because this is actually about the translational development now, and then we don't want to just use a random concentration of the inhibitor. We first ask ourselves that what are actually the concentration of this drug that were actually achieved in the patient who received this drug. And then so we asked that question first, and we found that the peak concentration in the peripheral blood is around 1.2 millimolar. And then because of this, then we actually carry out a dose titration experiment around this concentration. So we did push up a little bit up to 5 millimolar, but we also titrated further down to as low as close to 0.1 millimolar. And then here we used isolated CD4 to ask the question of whether this drug as sort of a physiological concentration, can it more selectively suppress the TS17 response? And to our surprise, the answer seems to be yes. So at 1.25 millimolar, sort of close to 1.2 millimolar, we observe a very, very nice suppression of the IL-17A and IL-17F, and with very, very minimal impact on interferon gamma production. And what's worth mentioning is that even if you reduce the concentration to 0.6, you actually still can see about 60% of the downregulation. And another thing that's worth noting is that when we push up the concentration to 2.5 millimolar, then you can actually see a completely removal of interferon gamma. And then so this is probably a concentration that we want to avoid, because apparently it starts to actually act as a sort of a non-specific T cell suppressor. And then this might actually lead to some of the unwanted adverse events, such as the increased chance of infection to virus or TB. And then so we were quite pleased with this data in T cell. But what about the monocyte? So we carry out something similar, and then we're quite actually pleased to see that in general, the potency of this drug in suppressing TNF alpha was as good as its suppression of the IL-17A in CD4 cells. And then we also observed similar level of suppression for IL-23. So I've highlighted this 1.25, this sort of more physiological concentration of the drug. But what's worth highlighting is that for some reason, actually, this drug do not affect the production of either IL-1 beta or IL-6. So we haven't really started to look into the biological mechanism of this kind of the pattern, but this is something that's quite interesting. It seems to be targeting some pathway that's actually shared by TNF alpha and IL-23, but actually not by this pathway or pathways are not affecting the production of IL-6 and IL-1B. So to summarize this part of work, so essentially we found that either IRE1 inhibition or existing drug can actually directly block three key elements of the AS pathology: 17A production by T cell, IL-23 and TNF-alpha production by myeloid cells. So here we mostly focus on the monocyte and macrophage, because we know that in the context of ankylosing spondylitis, those two cells are the main source of the TNF-alpha and IL-23. And so we're actually quite excited about this project, and then not only because of the biology revealed by this project, but also because actually through this project we know that actually our strategy set out in the beginning seems to be working. And so we use actually genetic-led research to identify an important immune regulator, and then based on this discovery, then we perform a translational research, and then we can actually identify FDA-approved drug that can actually relatively quick quickly be brought to patient. And then as lots of you might know, actually, the discovery of the development of a drug from a drug target to actually patient, it can easily take more than 10 years. And this will take such a long time. And then for us, we definitely would like to have our work have a positive impact to the patient's life as soon as possible. And because of that, I think that drug repurpose is very likely to be a very, very good approach that will definitely and rely on in the future. And then, so there are some ongoing work. So we are actually testing the in vivo efficacy of the IRE1 inhibitor and the drug X in murine model. And actually, we have already got some data. The in vivo efficacy of the drug X was actually not very good in murine models. And the main problem that we were facing was that it was really hard to achieve the concentration that magic 1.2 millimolar concentration in mice. But the IRE1 inhibitor actually seems to be suppressing the T cell inflammation, and then in some of the models suppress the arthritis inflammation as well. So we are still working on that. And then, as I said, that we are very keen to explore the possibility of performing a trial for the drug X in AS. But here I think we are facing some problems as well. Because the drug actually have to be given a very, very high dose. And then this is likely because, as you might see here earlier, that actually this drug has really a short half-life. And so we have been having some discussion with MedChem people. And then maybe one way to do it is actually to induce some of the modification that hopefully can enhance the half-life of the drug. And so if we can do that, we might be able to actually reduce the dose of the drug. And then once we solve that problem, then this could actually be a quite a nice things to proceed. And then after this, I think I would like to share with you of a few things that we are working on in the lab, or a few sort of techniques and methods that we are developing in the lab. And the first thing is that apart from this YAO1 gene, we have discovered two additional immune regulator in the myeloid cells, mostly in the primary monocyte. And also this is something that we are still working on at the moment. But when we are working towards our goal, we definitely experience and also appreciate the limitation of the siRNA stream. Because siRNA is transient, it normally lasts for like 3 up to 4 days normally. And so if we want to perform some of the longer assays, especially more complicated assays involving the organoid formation, and then starting the crosstalk of different cells, we might actually need to culture the cells for weeks. And then so in this case, siRNA is not going to work. And then apart from that, we actually also have some of the interesting and unexpected findings. That is that for one of the projects with our siRNA, somehow it increased the expression of its target rather than reducing it. So this is apparently will be involving the complex biology of how siRNA work. But one possible explanation is some of the siRNA stabilize mRNA rather than actually in facilitating the degrading of the mRNA by DICER. And then lastly, although pool screen has been carried out in cell line, so using the lentivirus shRNA, but for siRNA for primary cells, siRNA is not really an option. So because of that, we spend make lots of effort trying to actually set up the CRISPR knockout and CRISPR screen in the primary human cells. And then initially we tried to achieve a good efficiency of gene knockout in 3 major immune cells: primary CD4, monocyte, and macrophage, all of them from primary cells. And then we were able to achieve really high level of the knockout efficiency. So this was sort of the first milestone achieved. And then after that, built on those techniques, and we performed some of the a small screen and using the TS17 as a cellular readout, and so essentially what we did was that we firstly using a RMP. This is essentially a combination of the recombinant Cas9 protein with the synthetic guide RNA. So it has this complex can be delivered into the nucleus of the cells through the electroporation. So after this sort of electroporation, essentially this is knockout happens from here, and we give the cell 6 days for the endogenous protein to be degraded, and after that, then we place the CD4 cells in a sort of a TS17 expansion conditions, using both of the IL-1 beta, IL-6, IL-23, and the T cell receptor activation beads. And then we culture the cells for 6 days, allowing the TS17 response to happen, allowing the TS17 cell to expand. Then we look at the both of the cytokine production in the supernatant, and also looking at the frequency of the different subset of the cytokine producing cells, sort of similar to the experiment approach that we carry out using for the microRNA 10B project. And Feng is the DPhil student who has been leading this project. And so, sorry that I have to cover the name of the genes because this project still is of a more earlier stage, and we are not ready to definitely not ready to publish it yet. But hopefully, if you trust me, we have screened 1, 2, 3, 4, 5, 6, 7. We've screened 7 genes, and then all 7 genes are the genes that has genetic support. So that's linked to the AS-GWAS genes, and then we chose AS-GWAS genes because TS17 cells are known to be very important for the AS pathology, and then we thought that because of that, if we want to find if we want to provide evidence showing that actually the genetic-led investigation can identify new disease-relevant biology, would be best to utilize the genetics from AS and then use a cell that's relevant to AS, namely TS17, for this purpose. So that's why we chose to focus on AS genetics and focus on TS17 response. And what we found was actually just out of these 7 candidate genes, we found one of the novel regulator that's important for the TS17 cells. So on the top, this is the flow cytometry data showing you the frequency of TS17 positive cells. On the left, on the bottom is the level of IL-17 cytokine in the supernatant, and then you can see the knockout. So first column is the control condition. You can see that following the knockout of this gene, both of the frequency of TS17 cells and the production of IL-17A in the supernatant were downregulated. So this approach seems to work, and then importantly, the knockout seems to work really well in a sort of array screen. So this is we normally call this array screen because you knock out 1 gene at a time. So we call it array screen. And then if we actually knock out hundreds or thousands of genes at the same time, then we call it pool screen. So this kind of screen is really useful, and then especially particularly useful for the validation of the target. But the throughput is not very good. It probably to screen 7 genes, it will take us at least 1 month. And then so we like to have something that can give us a bit better throughput. And then and because of that, and then we decided to actually established a technique that was published a few years ago. So in this technique is called a slice. So essentially, what we carry out was the approach was carried out based on the combination of the lentivirus delivery and the electroporation. And the lentivirus mostly just a system is used to deliver the guide RNA, which essentially can guide the Cas9 protein to the site that you want the double-strand DNA break to happen. And then the Cas9 protein is delivered by electroporation. So this is sort of similar to the array screen method that I just showed you. So through the combination of these two approaches, you can actually achieve really high level of the knockout efficiency. And then importantly, this type of combination allow a pool screen. And in this example paper, and that established this approach, they were able to perform a genome-wide CRISPR screen in primary human T cell, which is really astonishing. And then so we have tried to achieve establish this technique in our lab. So we firstly try to do a sort of just for one target, which is the beta-2M, and then we were able to achieve about 5% of knockout efficiency. And then this is because actually we didn't actually concentrate the lentivirus. And so we were actually quite encouraged by this, because without any concentration, if we can achieve this level of efficiency, of knockout efficiency, then if we just concentrate the virus by say 10-fold, then we might actually get. And this is actually a typical knockout efficiency that you liked for a slice like genome-wide CRISPR screen. And then I'm going to skip this. And then for this project, for this sort of a technique, now we recently have a new staff join the lab who have been doing the CRISPR pool screen in the past, and he is going to work with some lab members, including actually Pablo, who most of you already know, and to perform this work. And then so because this method actually has been quite very nicely described and has actually been used. So we thought that this should be something that's easy to carry out, and we're confident that we can do that. And then we move our focus towards the human macrophage, because we want to do this for at least both of the T cells and then the major myeloid populations, including monocyte macrophage and DC. And because of that, then we want to set it up for the human macrophage as well. But we found that actually only 2 publications that describe the genome-wide CRISPR pool screen in the macrophage are actually either based on the cell line on the first paper, or based on iPS-derived macrophage. And this is not ideal, because again, I think I cannot highlight it enough that the cell lines are different from primary cells. Wherever we can, we should just directly work on primary cells. And then so if it's not available, we would like to develop it ourselves. So we firstly try to actually sort of learn from this slice approach, and then try to see that if we can actually use lentivirus to deliver guide RNA to macrophage, and then followed by electroporation to deliver Cas9 to macrophage. We've tried that, but we found actually macrophages are very resistant to electroporation. The Cas9 do not really enter the differentiated macrophage there well, and so this is not a very good option. And then we start to actually develop some of our own approach, and then we try different methods. And one of the methods seems to be working. Is actually to combine the chemical reagent-induced Cas9 deliver with the lentivirus assisted guide RNA delivery. And using this approach, now we can achieve about as you can see here, about 50% of the knockout efficiency. And then, but actually with a lentiviral system, we still need to improve the efficiency of the guide RNA delivery. So currently we only have about 2%. And then, so we are working to increase this percentage to about 50%. And then once that's achieved, then we are quite confident that we should be able to establish a method for the CRISPR pool screening in the primary human macrophage. And again, Max is working on this project. And then, I think I might skip this part because I would like to save some time for the discussions. And then this is just some of the additional kind of the a method that we would like to develop. And then so in summary, oh yeah, there's another part. I might skip this part. So in summary, we have been built on we have been sort of building and optimizing a strategy for us to combining different data set, different omic genetics data set, omics data set, and then powered by the either pool or CRISPR pooled or array CRISPR screen to identify new biology, and then based on this kind of the sort of a novel biology or novel immune regulator discovery method, and then we can then perform, we can validate the target, and then we can perform the mechanism, perform studies for the mechanism or for the translational development. And then for the genetic risk, we currently, I think we use the eQTL dataset more, and then but definitely apart from eQTL, there are lots of other options that can be utilized, including like pQTL, and then including the sort of prompting looping, so prompting 3D interaction kind of a data set. And then those data sets, once they become available, I think we'll definitely be keen to integrate that kind of data set into our sort of approach. And then apart from for the gene, and so for the function, we have been mostly rely on the omics to predict the potential novel regulators, and then this omics actually come from this part, which we will talk about a bit later. And then finally, for validation, we use siRNA and CRISPR. We haven't completely give up siRNA because in some cases siRNA is useful, especially for macrophage. We thought siRNA is actually more user friendly than the CRISPR. And then for the array screen, yeah, I think we have used array screen quite extensively. We can do that for both of the T cell and the macrophage, and then we are very close to establishing the pool screen, and then we are looking forward to some of the data that might come out of the pool screen, and then we were very keen to actually after we establish the pool screen, then we will be actually focusing on establishing the intercellular CRISPR pool screen and the single cell CRISPR pool screen, and that will actually provide even higher power, especially for the 3D kind of the in vitro modeling system. And for the omics data set, I think now we found the patient tissue single cell data set is most useful and powerful approach. And then especially this type of data set when for the condition for the disease where actually the data set is available from both of the patient group that responds to current treatment and the patient group who do not respond to current treatment, then this kind of data set could be really useful and important because it might actually reveal a novel biology that explain why some patients do not respond to current treatment. And if we understand why, then we can actually try to address those potential pathways or pathologies that are actually unique to those population of the patient that really need some help and new drugs. And then we found that disease and tissue specific cell subset omics will still be useful. Sometimes this sub data set might not be as powerful as a single cell But in the cases where single cell data set is not available, then this type of data set will still be useful. And then, as you have heard, actually, we indeed use this type of data set to identify the ER1 gene as an interesting candidate to take forward. And then lastly, and this is, I think, a very, very challenging bit. Everyone thinks that this is really important, but it's really difficult to do. That is how to integrate the single cell and omics data with the GWAS data. I think the integration, especially high-level integration, is really, really challenging. And then I think this is something that we are working on at the moment. And then the data set such as the single cell ATAC sequencing. ATAC sequencing is kind of a sequencing method that allow us to identify the genomic region that are actually open to the binding of the transcription factor, and then often those regions are more likely to be transcribed at a higher level. And so if we can generate the pair single cell RNA sequencing and single cell ATAC data set, then we can use the RNA sequencing data to actually guide us to the important population of the say monocyte that's important for the treatment failure, and then if we then look at this population of the cells, and then look at the ATAC sequencing data set, and then ask the question that in this population of cells, whether certain genomic region, especially the genomic region that are actually overlapping with the genetic risk, whether those regions are actually opened. And then this kind of analysis can really provide very, very useful and then very, very sort of intelligent approach to integrate the genetics data with a single cell. But this is something that we are still optimizing, and I think we'll probably still need lots of work before it can be actually used to help us to discover a new biology. And then, yeah, for the translational project, I think I've told you about the IIU1 project, and then we also have another project about the PMT6, and this is a project that has gone really, really far, and we even got the venture capital fund to perform some of the really preclinical and development. And then, but it is another very, very complicated story. I think hopefully I'll have a chance to show you next time. And then, lastly, yeah, I should really thank everyone in my lab. Apparently, I didn't do the work; they have carried out all those amazing work. And then I should thank all my collaborators, including Paul, based in Oxford, Richard William, based in Oxford, Julian Nye, based in Oxford, and Duan and Hai, based in China, and then thank all the funding bodies. Thank you so much. I'll stop here, and then looking forward to any questions.
Yeah, thank you very much, Llye, for your talk, and thank you again very much for accepting to collaborate with us with this amazing talk. So there are some questions from the public, yeah, and first of all, Dr. Tournier wanted to thank you for your presentation and to say that your career has been very impressive, and he wants to congratulate you for the integration of the immunological knowledge that you have.
Thank you.
So that's the one. That's the first one. Okay, and then I have another question from the chat, which says, is there evidence of gut inflammation in the models that you have used, or any evidence that proves the existence of a gut joint axis in the spondyloarthritis?
Yeah, thank you. I think that's really a very, very good question. And then the sort of the gut-driven information. I think everyone will agree that it must be a very important aspect of the pathology in ankylosing spondylitis. And then actually this is the one of the very last project that I skipped actually was addressing this project. And so essentially, firstly, apparently for in vitro model, we cannot possibly address the biology related to the gut and then gut inflammation and traveling to the through blood and then contributing to joint inflammation. This is something that we can't do, but we were able to do through that project was that we found that some of the subset of the TS17 cells, they actually have the characteristic of the stem-like cells, and then those cells, not from our work but from mirroring work, there were two studies published in Nature about 2 years ago, and then and firstly, one of them showed the presence of this population of the cells in the TS17 compartment. And then the second paper shows that those population of the cells were actually generated in the gut, and then they were sort of maintained in the gut, and then serve as a sort of a reservoir for the generation of pathogenic TS17 cells. And then in that paper, they also indirectly suggest that the pathogenic TS17 cells were also actually generated in the gut, and then only for certain subset of the TS17 cells having the certain combination of the chemical receptor, they were able to get enriched in the inflamed tissue. So in the model, they were not starting arthritis; they were starting the neural inflammation. So they were showing that a subset of the TS17 get enriched in the inflamed brain. And then so what we have been doing was analyzing the paired single cell RNA sequencing with the single cell T cell receptor sequencing, and then using this kind of approach, we were able to sort of compare the T cell receptor usage between the TS17s, between different subsets of TS17s first, and then secondary between the TS17s coming from the blood versus the TS17s that is located in the joint. So this kind of approach is quite powerful. And then one of the for that project, we have analyzed some of the existing data set that's available from Paul's lab, and then but the cell number was quite too low, and then so we have carried out a separate experiment increasing the T cell numbers by 5 times, and then we are still waiting for the result. And so I think to sort of sum up and about your question, I think for the in the context of the human based work, I think we will have to rely more on the cells from patient, and then to understand to help us understand a bit better about the gut and joint axis. Now I think we really need to actually rely on the combination of the RNA transcriptomes with the T cell receptor usage, and then but if we want to actually perform some of the in vivo functional work, then I think we do have to go back to the mouse model such as SKG is probably the best mouse model for the AS, especially for the gut and joint axis topic.
Okay. Thank you very much.
Thanks.
That question was from Brian. And then I have another question from Ricardo that says, thank you for your excellent presentation, Doctor. Why the interaction between HLA-B27 and IRAP1 seems to be reflected in articular inflammation and not in systemic inflammation? Yeah,
I think that's that's sort of I think that's a very good question. But at the same time, I think maybe let's take the psoriatic arthritis, for example. So in the psoriatic arthritis, still a good proportion of the patient are positive for HLA-B27, but there are also a substantial proportion of the patient that are not HLA-B27, and then for this group of the patient, it's very likely that the T cells. Earlier we were talking about T cells educated in the gut and then messing up the joint. But in the psoriatic arthritis, some of the T cells can actually get educated in the skin, and then those cells can mess up the skin, but also mess up with the other organs, including joints. And then I think in terms of the HLA-B27, I think it's very likely that this is probably very closely linked to the gut inflammation. And then I don't know whether you are familiar with the literature in the gut dysbiosis in AS. Matthew Brom's lab, I think it was in 2016, had work in the arthritis and rheumatology, showing that when they compare I think they used the shotgun sequencing at that time, and then they compare the microbiome of the B27 positive and negative healthy control, and also B27 positive and negative AS patient. They found that even in the healthy control population, the presence of the HLA B27 affect the microbiota. And so that's sort of a very strong evidence suggesting that it's possible that the presence of the HLA B27 itself can predispose the human beings to ankylosing spondylitis through the alteration of the microbiota. And then, but I think for this, I think we still need to wait for more data to come out. And then, and again, I think I cannot emphasize enough. I think for this kind of question, we should really focus on the data coming out from the primary human and primary patient samples, because that's actually most relevant to our research question. Thank you.
Good. Thank you very much. And another question that comes here is, when you inhibit IRE1, is it possible that at the same time you are inhibiting the physiological functions of endoplasmic reticulum stress?
Yeah, I think that definitely. So as I sort of touched on earlier in the talk, the IRE1 and XPP1 pathway is an ancient and it's a very, very important pathway for many of the biological response. So that's precisely why that we had a concerns about targeting and using IRE1 inhibitor to target IRE1. And then, but actually, with that being said, what I didn't say earlier was that there are actually 2 clinical trials that's going on at the moment, testing the efficacy of IRE1 inhibitor in treating solid tumor and the lung fibrosis separately. And then both of them are phase 1 clinical trial. And so we are actually very curious about the safety profile from those 2 trials. And so if actually the safety profile is actually better than we expect, then this could actually be a good option to take forward. And I think something relevant to this topic is that back in, I don't know, maybe 20 or 30 years ago, when people start to think about targeting TNF-alpha to treat arthritis, everyone thinks it's crazy because TNF-alpha is so important. And then how can you target something like that? And then similarly for the JAK kinase inhibitor. JAK kinase is important for so many vital physiological response. And then you'll be actually very worried about the safety profile targeting of the drugs targeting those pathways. But the fact is actually it's all right. There are definitely some adverse events, but the safety profile seems to be actually much better than we thought. Now I think the key potentially a key element is actually those inhibitor is not as effective as knockout. So they only actually reduce the activity of those pathways, and especially I think JAK kinase inhibitor. So if we tune down those pathways rather than completely knock out those pathways, then actually we might be all right.
Okay. Thank you very much. And this is the last question. It says, thank you for the presentation, doctor. Which do you consider that are the principal or the main osteogenic molecules implicated in spondyloarthritis, and how did you couple your results with neobone formation?
Yeah, so the first question, I think we still don't know. And as I said, I think the biology of the even just HLA-B27 biology in AS is still not clear. And then so I think what is clear in AS is that definitely there's increased IL-23 response in the peripheral monocyte. and then also peripheral macrophage has been shown by many people. And then it's very clear that the IL-17A producing T cells are increased in the patient, and then the blocking of IL-17A is effective in treating those patients. And then the blocking of TNF alpha is effective. Interestingly, the production of the TNF alpha in AS is not necessarily upregulated. There are some controversial there. Some research shows that these macrophage and monocyte from AS patient produce higher level of TNF alpha in response to stimulation, but others actually shows no difference. So I think, but for some reason, actually TNF alpha work very, very effectively, at least for almost half of the AS patient in controlling the conditions. And so listen, I think this is what we know. And then about AS, and then so hopefully in the with time, hopefully we can actually know better about the pathology of ankylosing spondylitis. And actually, as I sort of also alluded earlier, that in our lab now we are actually revisiting some of the previous work about Llye and about HLA-B27. As I said, I'm not completely convinced by own data, and so I would like to know that whether this kind of the finding were correct in the in patient or not. And then we have at least 2 projects going on at the moment, trying to understand a bit better about how Llye and HLA-B27 work in the context of the AS. And then yeah, hopefully maybe I think we probably need another 2 or 3 years before we can generate very clear data to answer those questions. And about newborn formation, again, I think this is a very, very good question. And then I have to be honest that we as a field hasn't really been putting enough effort in understand the newborn formation. And especially if we're thinking about comparing the rheumatoid arthritis with ankylosing spondylitis, they are so different, right? Rheumatoid arthritis bone actually get eroded, and then in AS it's opposite. There's newborn formation is one of the sort of key feature of the AS. And then for both of the condition, TNF alpha apparently is important for both of the conditions. IL-17 is important for AS, but not important for the rheumatoid arthritis. And then and I think what is probably missing is actually the stroma cells, and because talking about newborn formation and talking about the bone generation, about I think 2 or 3 years ago there were 2 sequential Nature paper focusing on rheumatoid arthritis, describing the presence of the 2 main subset of the fibroblasts in rheumatoid arthritis, and namely the lining and sublining fibroblast. And then this lining fibroblast is actually more important for the bone erosion in rheumatoid arthritis. And in contrast, the sublining fibroblast is more pro-inflammatory and supporting the T cell and myeloid cell inflammation. And then one would actually expect that the pro-inflammatory subset of the fibroblast, the sublining fibroblast, are very likely also actually expanded in the patient with ankylosing spondylitis. And then those lining fibroblasts is very likely actually not active in the ankylosing spondylitis. And then if we're thinking about newborn inflammation, it might actually go other way around. And then the last thing that I would like to highlight is that I think there was some initial work showing that the stem cells, the stroma located stem cells might be relevant to the newborn formation. But I think that kind of the data is still, I think, only came from a limited number of the research labs. I don't think we are completely convinced that the sort of tissue resident stem cell like cells or the progenitor cells are contributing to the newborn formation. So yeah, still I think I don't have clear answer for either of your questions. Sorry.
Okay. Yeah. Thank you very much. Thank you very much, Llye, for being here today. And I think we are in time now. So thank you everyone who assisted to the conference. See you soon. So thank you, Llye. Take care.
Thank you. Thanks,
Thank you, everyone.
Thanks, everyone. Bye-bye. Have a lovely day.
Bye.
And enjoy the rest of the conference.
Yeah, thank you very much. Have a good time. Bye-bye.
Bye.
Les agradecemos mucho por su asistencia. Al punto de las 11:30 seguimos con nuestras transmisiones en vivo. Muchas gracias.